Hello everyone, and welcome to our webcast event. My name is Brandon Williams, and I'm a Global Product Manager here at Bio-Rad Laboratories. I also have here with me Dr. Chris Brampton, who is our flow cytometry application scientist, and we will be your moderators for the following presentation. So I think everyone would agree that single-cell RNA-Seq is an incredibly powerful tool and has been instrumental in helping researchers understand how rare cell populations, as well as the behavior of individual cells, shapes biological processes. And with the introduction of turnkey, droplet-based single-cell analysis platforms, more and more researchers are turning to single-cell RNA-Seq to uncover these biologically important rare cell populations that have been shown to play critical roles in the pathogenesis of cancer, mediating immune responses, and drive the development and repair of tissue. But what if the goal is to analyze the cellular heterogeneity of a cell population that is itself already rare? Our presentation today will detail work that demonstrates how pairing high-speed fluorescence-based cell sorting with single-cell RNA-Seq makes it possible for researchers to find success when undertaking this challenging endeavor. Now, before I introduce our speaker, I would just like to quickly highlight that we here at Bio-Rad have developed our own very approachable and incredibly easy-to-use benchtop cell sorting system that was engineered for the isolation of rare cell populations. The S3E cell sorter not only sorts cells at very high speeds, but it does so gently, so you can feel confident that you're maximizing your single-cell library prep and sequencing results by only using samples that are highly viable. If you would like more information on the S3E, you can follow the link at the bottom of the slide. I would also like to take this time to highlight that this is an interactive webcast, and so if you'd like to ask a question, there should be a Q&A box located on your screen. Simply type in your question into the box, and we will try to get to it after the conclusion of the presentation. If, however, we run out of time, we will make sure to follow up with you via email. And with that, I'd like to introduce Dr. Bassal, who will be discussing some of his research in his presentation titled Single-Cell RNA-Seq of CD34 positive, CD45 positive, sorted, unstimulated human peripheral blood reveals cellular heterogeneity within HSPC pool. Dr. Bassal has a dual appointment at both the Beth Israel Deaconess Medical Center at the Harvard Medical School, as well as the Cancer Science Institute of Singapore at the National University of Singapore. Dr. Bassal earned his PhD at the University of South Australia investigating germline variations that can give rise to the altered metabolic phenotypes observed in adult acute myeloid leukemia. Currently, he works between the Harvard Medical School and CSI Singapore, investigating healthy and tumor microenvironments at the single-cell level, in addition to broader research efforts to better understand DNA-RNA binding protein biology. Thank you, and I hope you enjoyed today's webcast. The floor is yours, Dr. Bassal.
Hello, and thank you everyone for attending today. And special thanks to Bio-Rad for giving me the opportunity to present the work that has been done on this project so far. So I wanted to start with a bit of background. So over the years, a significant effort has actually been made to pull out and characterize hematopoietic stem and progenitor cells, both from bone marrow, peripheral blood, and cord blood. And this was then using a number of strategies, including antibody marker panels, FACS, transplantation, biochemical assays, and so on. And so a great deal has been done to better try and understand hematopoiesis and its differentiation hierarchy. Now, with the increasing use of single-cell RNA-Seq, researchers have sought to further our understanding of hematopoiesis, and many studies have been published investigating the differentiation hierarchy, both in humans and in mice. So in this paper from the Bassal lab, they were able to construct a really nice hierarchical tree of stem and progenitor populations. And other studies such as this have really tried to push the bounds of our knowledge by better characterizing the different lineage paths and differentiation trajectories that are found in biology. For our study, though, we really were interested in trying to better understand the population right at the top of this tree, the hematopoietic stem and progenitor population. So to quote some numbers here, in human peripheral blood, leukocytes are about 1% of circulating cells. And within this 1%, the stem and progenitor population makes up between 0.03 to 0.09%. So very abundant, as you can see. Now, the literature adds a little bit more detail in that if we look for the same population in bone marrow, these cells are about 10 times more prevalent. So still within that, quote unquote, super prevalent range, as you can see. So we set out with a very simple aim in this project. And that was to investigate the transcriptional landscape of the stem and progenitor population at the single cell level. And we wanted to do this by starting from a sorted, pure cell population. And the reason for that is you can sort a cell population and be confident by facts that it's pure, meets the right markers and everything. And then you sequence that using bulk type approaches. The thing is though, since single cell RNA-seq became a thing, we're learning that even with quote unquote pure cell populations, once you profile them at the single cell level, you can see heterogeneity that was previously hidden. And this can be attributed to the diversity of cells and the differences in transcriptional programming inherent to different populations that may not be reflected at the cellular surface in a manner that will allow differentiation by facts. But even within a sorted pure population, heterogeneity can still exist. And how detailed a picture you can draw at the single cell level is really affected significantly by how well your purity selection step was. So what we did was to start by sourcing fresh, unstimulated peripheral blood mononuclear cells from healthy non-diseased donors. Next, we used the Bio-Rad S3E and selected for CD34 positive, CD45 positive cells. The cells that were pulled out were then encapsulated using the Bio-Rad DD-Seq platform and library generated as per the kit instructions. The samples were then run on an Illumina NextSeq, with demultiplexing being done using the base-based SureCell app, with subsequent analysis, filtering, and normalization being done using an in-house R pipeline. To visualize and to be able to interact with the data in real time, I used the Spring program, which was developed by the Klein Lab at the Harvard Med School. Finally, the bioinformatics work is great and all, but the validation work is coming along nicely with some exciting findings to hopefully share with you guys in the near future. Okay, so for those of you that were interested in the gating strategies that was used, the PBMCs were stained with CD45 FITC, CD34 PE. Forward scatter and side scatter gating enabled exclusion of debris and cell aggregates. 7-AAD staining identified dead cells. And so in the end, CD34 positive, 45 positive, 7-AAD negative cells were isolated for single cell RNA-seq. A post-sort purity check was found to be 97.8 pure with 98.4% cell viability. All right, so 2 replicates were actually run. Now, after demultiplexing, cells with more than 200 detected genes were selected, and this gave a starting cell number of 1,914 cells. So looking at the library size, so how many cells have how many reads, and the number of expressed genes, we start to see what properties the cells in the cell population actually have. In this case, it's pretty clear looking at the histograms that there are outlier cells that don't fall in line with the ideal profile curves that are actually shown. Also, looking at the mitochondrial percentage, you can see again that there are a few cells that just don't fall in line like the rest of the population. So once you do some trimming, voila, we have our cleaned up data set for further analysis. And in the end, there were 1,708 cells that passed all the QC, all the filtering, and the quality thresholds that were set. After looking at the overlap between the 2 replicates, there were no significant batch effects that were found to take into account or to account for. So the data set was really in a good state to move forward with and analyze further. So moving on to the visualization and interaction with the data. As I mentioned previously, I used the Spring tool, which was developed by the Klein Lab. And what this does is it generates a graph of the cells by running a principal component analysis on the cells in gene expression space. And then it generates a k-means clustered k-nearest neighbors graph and plots that in 2 dimensions, which you can play with and interact with in real time. Differential gene expression was performed in R, and pathway analysis was done using the QIAGEN IPA toolkit and suite. Now, I want to touch briefly on the point of dimensionality reduction in single-cell RNA-seq. I've mentioned a couple of times that for this data set, I use principal component analysis. And typically in single-cell RNA-seq papers and publications, you will see t-SNE or UMAP plots presented. And the choice of technique really has a major effect on the interpretation of the data and so potentially really on the outcome of the project. So simplifying a few details and giving you, rough rules of thumb that I like to go by. Principal component analysis preserves the intra and inter-sample relationships, but it does so at the cost of separation between the groups. TISNE, on the other hand, it sacrifices that inter-sample relationship for the sake of separation. Finally, UMAP attempts to find that middle ground between principal component analysis and TISNE. So I have a personal preference for principal component analysis, and the reason I feel this way is that I see that biology is a continuum, and so you may not necessarily have discrete populations, and cells tend to, change phase from one quote-unquote cell type to another quote-unquote cell type. So cells are constantly transitioning, from phase to phase to phase, and you don't see these distinct, discrete cell populations. Obviously, this is really dependent on the data set that you're working with and on the cells that you're investigating. So some cell populations will actually, separate out nicely with principal component analysis, and other cell types, for example, neuronal cells in particular, principal component analysis just fails in that regard. And so you really need to use something like TISNE or UMAP to get that great separation between the clusters that you're actually investigating. So taking the data set that we have and running a PCA on that, what do we see? So this is the spring representation. Now here, every circle represents a cell. And the different colors that you actually see represent clusters of comparable expression profiles. And so what we see here is the analysis came up with 10 transcriptomic clusters, each in its own color. Now this number of clusters, so these 10 clusters, it's not a number that was just arbitrarily chosen or anything like that, but rather looking at the analysis and seeing how many significant principal components can, explain the data. And this number was used to drive the number of appropriate clusters to show. Now, despite the algorithm suggesting that there are 10 transcriptomic clusters, careful examination of the data, I was only really able to figure out and decipher 7 major biological populations of interest. And so just to make it easier and clear what I'm actually referring to, I'm going to temporarily and artificially just, pull apart these different populations so you can get a much better understanding for what I'm going to be referring to over the next couple of slides. So you can see we've got population 1 to the left, and then moving to the upper arm, you've got populations 2 and 3. And then going towards the lower arm, we've got populations 4, 5, 6, and 7. Now, once I separated out the clusters, performing differential gene expression of each population and then subsequent pathway analysis revealed which cellular pathways show, predicted activation or deactivation. And this can be seen in the bottom left-hand corner here, where green represents pathways that are predicted to be deactivated, moving up to purple, in which pathways are predicted to be activated. And what we can see is that population 1 shows many cellular pathways in a predicted deactivated state. And there were only really 3 pathways that appeared to be activated. I'm showing 2 on the slide here. Whereas the rest, which is easily over, 30, 40, 50 pathways, show predicted deactivation signature. What's interesting, though, is that once you start moving towards the right, you see cellular pathways activating, suggesting cells are progressing from a dormant to a more activated state. So this really, suggests that the cells appear to be maturing or activating, through different phases. And this is consistent with, activation of multiple cellular pathways. So, bringing and collapsing everything back together again, how it's actually meant to be viewed, we see the populations in their state. So we've got, population 1 moving up to populations 2 and 3, and then also population 1 progressing to populations 4, 5, 6, and 7. Knowing this, though, wanted to dive a bit deeper into this data set and really wanted to see what can we learn about each of these different cell populations. First up, the first population all the way to the left. In this population, there were no genes that showed a unique expression profile just for this population alone. Differential gene expression and pathway analysis showed a signature that is consistent with downregulated gene expression with respect to the other cell populations. And this suggests deactivated cellular pathways, as I discussed just a couple of slides ago. Gene set enrichment analysis also showed signatures that were consistent with this observation of, downregulated and deactivated cellular pathways. So visually, what this looks like on pathway plots from IPA. So in relation to EIF2 signaling, and so this pathway, really outlines the different participating members associated with ribosomal activity. And what we can see is that many of the members in this pathway, show green, which means that their gene expression is actually down regulated with respect to the other pathways. Looking at oxidative phosphorylation, we see a very similar picture going on here, that many of the genes that make up these 5 different complexes also show downregulation in their gene expression. Moving on to gene set enrichment plots. Again, we see strong signatures associated with downregulated peptide chain elongation, ribosomal activity, and translation. So taking all of this information together, we really suspect that this population is in a transcriptionally inactive state. Now, with hematopoietic differentiation, for those of you who are not familiar with the details, here is a simplified schematic of the classical differentiation tree, on its side, taking a break. And we have an early branch point which separates out the myeloid and the lymphoid lineages just after the multipotent progenitor stage, the MPPs. This separation is marked by, expression of distinct genes, but FLT3 and GATA1 can really separate out these branches. And in our data set, we can clearly see a strong separation between the 2 regions of this population, with FLT3 expression being enriched in the bottom half of the population, and GATA1 being enriched in the upper half of this cell population. So moving on to the second population, we see genes that are associated with commitment to erythrocytes, such as CSF2RB, which is IL-3 receptor. Now, what is known is synergism between the erythropoietin and IL-3 occurs in the induction of hematopoietic stem cell proliferation and erythroid burst colony formation. CNRIP1 is one of the few characteristic genes known to regulate erythrocyte and megakaryocytic differentiation. ITGA2B, KLF1, and HBD are all also associated with megakaryocytes and erythrocytes. So these are, what you can see on the slide here, are representative spring plots showing which cells show the expression of which genes with what intensity. Now that you can see what the data would actually look like on a spring plot, for the rest of the presentation, though, I'll only be talking about the genes that are actually expressed rather than showing you individual spring plots for each gene that I'll be talking about. Finally, moving on to population 3 in the upper right portion of the cell population, we see further evidence of erythroid commitment with APOC1, which is found upregulated in cells undergoing megakaryocytic erythroid differentiation, CD36, which is found to be associated with megakaryocyte erythroid-committed progenitors, UCA1, whose expression is dynamically regulated during human erythroid maturation, with a maximal expression in proerythroblasts. So you can see that as we've moved from population 1 up to population 3 in that upper portion of the cell population, we do see signatures that are quite consistent with erythroid megakaryocyte cell populations. Going back to our differentiation tree though, now I've shown you evidence suggesting erythroid commitment in the upper half of the profiled cells. And so over the next few slides, I'll be talking about that bottom half, talking about the cells that show enriched expression of FLT3. Okay, so moving on to population 4. Now, this population is a bit of a tricky one. Firstly, there were no genes that showed a unique expression to this group of clusters alone. However, there was enrichment in the genes such as SELL, which is a leukocyte adhesion molecule, ANXA1, which is associated with hairy cell leukemia. So again, lymphoid lineage. And then SPINK2, which is found in a subset of lymphoid cells that's also associated with lymphomas. So despite there being no distinctly unique expression profile for this group of clusters, taken in the context of the assayed cell populations, it really does look like this population is an early lymphoid committed population. Moving further to the right and onto population 5, these look to be slightly more committed lymphoid cells with genes associated with such a population. So we see, enriched expression of LTB, which forms hetero-trimeric complexes on the surface of lymphocytes. We see MME, which is a membrane glycoprotein and a common ALL antigen. We see DNTT, and the encoded protein of which is expressed in a restricted population of normal and malignant pre-B and pre-T lymphocytes during early differentiation. And then finally, we also see enriched expression of J-chain, which is a protein typically associated with B-cell activation and differentiation. Next up, we're looking at population 6, which is just in the middle all the way to the right. Now, this population, there's evidence of myeloid-committed populations with expressions of genes such as MPO, which is synthesized only in myeloid and monocytic cells. And this is actually a really important marker for the myeloid lineage. We also see KBTBD11, which is a CML-associated protein. We see CDCA7, which is overexpressed in CML. We also see TNFSF13B, which is specifically expressed in monocytes and macrophages. And then finally, there's also enhanced expression of CTSG, and this has been associated with monocyte progenitors as well. So it looks like this population all the way to the right, based on gene expression signatures, could be a myeloid-committed population. Finally, moving on to population 7, all the way to the right at the bottom, this population really looks to be that it could be, committed dendritic cells. And the expression data would, lead us to that conclusion. We see enhanced expression of IRF8, which regulates the development of dendritic cells. And we also see expression of ITGB7, FGL2, and FGR, all of which are genes that have been, previously reported in the literature to be associated with dendritic cells and dendritic cell maturation. So bringing everything together from the last few slides, we see something that looks like this, which I believe is rather nice and neat. So here, the principal component analysis representation, I believe, is really, maintained and preserved the underlying biology, and is not shown, distinct populations floating around not connected to each other, but it's rather, maintained the underlying structure that we'd actually see or we expect to see based on the underlying biology. We see the different populations and how the cells sort of transition from one phase to the next. And we, begin to see differentiation paths as the cells become more and more committed. Also, the principal component analysis representation, I believe, has preserve the intermixing and the organized structure that we actually see here. Now, all of this was exciting, and it was really great to see this in the data, but there's still more that could actually be done. And so to dive a bit deeper into these cells and get a better understanding of how they're behaving, I wanted to do what was called an RNA velocity analysis. Now, I get that the term RNA velocity might be new for many of you, so I'm just going to do a quick 101, quick highlights of what RNA velocity is and what it actually tells us about the underlying cell population. So we all know that different subsets of genes are expressed in cells at different times, and the rate of expression can vary, per gene over time. And so after transcription, RNA gets spliced, at least in eukaryotes. And so nascent transcripts will have their introns still present. Mature transcripts, on the other hand, have had their introns removed, and they eventually get degraded. So if gene expression is actually increasing in a cell, you will see an increase in the abundance of nascent transcripts before the abundance of mature transcripts. So you'll see an increase in the number of transcripts that have their introns present before you see an increase in the abundance of mature transcripts where the introns have actually been removed. Conversely, if gene expression is actually decreasing, there will be a drop in unspliced transcripts before the spliced transcripts decrease in number. And so this ratio of spliced to unspliced transcripts present in a cell provides a glimpse into the future state of a cell's transcriptional state. More precisely, this is called RNA velocity. And so RNA velocity is the first time derivative of the spliced mRNA abundance. And so we can use this ratio to more or less predict and infer the future state of a cell. So in this great paper that was published by the Karchenko lab, I highly recommend that you actually go and read the paper. And they go into great detail as to how they were actually able to show that this is the pattern that you actually end up seeing in cells. And so again, by looking at this spliced to unspliced ratio, we can start to see or predict what the cell is going to be doing in the near future. So applying an RNA velocity analysis onto the data set that we actually profiled here, this is what we end up seeing. And so it looks a little daunting, so let me walk you through it. So first up, we have population 1. And what we can see is there is static motion straight through the middle of this population. And this is further evidence for that transcriptionally inactive signature that we saw and discussed before. The thing is that with population 1, we do see movement around the peripheries, probably suggesting a priming of cells to both cell states. So we see, arrows directing towards the upper part of population 1 and towards the lower part of population 1. Moving up in the middle towards population 2, we see some motion from population 1 to population 2. And then we also see a semi-stationary phase within population 2. And then finally, there's also movement away from population 2 to population 3. So this trajectory matches what the differential gene expression revealed, and the detailed investigation of the different populations. So the RNA velocity also suggests the same thing, that we're seeing cells, moving from population 1 up that upper arm towards a different cell state. Now, I'm going to skip population 4 for a tick and move on to population 6, where we see clear movement to a stationary phase. That's also the same for population 7 and population 5. We see, very strong movement to stationary centers within each of these distinct populations. Now, coming back to population 4, we see lots of movement here, each to the respective transcriptional cluster. So while there are 4 subclusters within here that I grouped together into a single population for the sake of my analysis, there does appear to be, transcriptional motion within these clusters. And this is probably why the principal component analysis actually suggested that you have different transcription groups within this major population. So even though I was unable to decipher exactly what was going on, there's clearly subpopulations that are actually present here. And so this is definitely warrants, further investigation and further interrogation of what's going on here. What are these different populations? Are there any, unique characteristics associated with these transcriptional groups? So it's interesting, but raises a couple of questions as to what's going on exactly there. So with all of this said and done, from everything I've presented today, what are the take-home messages here? So first up, within the sorted pure HSPC population, we can see a heterogeneous complement of cell populations that are identified, and each has its, very distinct, very unique transcriptomic signature. Also, the sorted ultra-rare CD34, CD45 PBMC population, while being, stem and progenitor cell population, does show differing levels of commitment to all the different hematopoietic lineages. And this is evidenced by the distinct characteristic gene expression profiles that I've spoken about in detail today. Finally, the heterogeneity of the HSPC transcriptomic landscape has really been revealed by combining single-cell RNA-Seq with cell sorting. So cell sorting has, given us the ability to get this really pure population. And then single-cell RNA-Seq has given us the ability to really dig down and look at the true heterogeneity within this population. Okay, so with all that said and done, I'd like to put a special thank you to the Acute Leukemia Lab and RNA Biology Lab where I work. Special thank yous to Dr. Cici Chen and Professor Daniel Tannen for their help with this project. Also, I'd like to thank the great people at Bio-Rad, Drs. Brandon Williams, Chris Brampton, and Alan Watson, and also the cell and digital biology groups. And then finally, a special thank you to Dr. Kelly Kaihara for her help in the start of this project. Thank you for listening, and I'm happy to take any questions anyone may have.
Hi, everyone. Thank you for joining our webinar, and a special thank you for Dr. Bassal for presenting his work. And it looks like we have some time for some questions. The first being, if on 200 of so genes expressed per cell, then are you, it looks like omitting the moderately and low expressed genes? Also, of those expressed, how many do you have sufficient coverage to look at splicing?
Okay, so to answer that question, the selection of picking out cells with, a minimum of 200 genes expressed, this was simply a starting point for the filtering. From there, what we do is look at the population of cells and determine what the average number of genes expressed per, across the population are. And from that, determine effectively deviation from that median, and we use that as a cutoff. So it's not that we selected 200 genes specifically, moving forward. It was just a starting point because we know that any cells with less than 200 detected genes, as a starting point, are just not going to be of any relevance biologically. In terms of those cells that actually have expressed genes, do we have enough sufficient coverage to look at splicing? I don't think that we actually do, keeping in mind that there's a 3' bias with the library kit. So, unless your splicing event is towards the 3' end of the transcript, you're not actually going to be able to pick it up. So you just got to be aware of, be it you're using the DD-Seq or the 10X or the InDrop or whichever platform is actually, used. You just need to be aware of the limitations of that platform and whether there's a 3' or 5' bias based on the kit.
Okay, thank you for that question. And the next question we have is, how many cells are the minimum to do the analysis? And if the cells came from diseased patients, how many patients are representative?
Can you repeat the question?
Yes. This question asks, how many cells are the minimum to do the analysis? And if the cells came from diseased patients, how many patients are representative?
Okay, so in terms of how many cells are minimum to do the analysis, it depends on how heterogeneous your population is and you expect it to be. In this situation, because we started from a sorted, relatively pure population, so we were able to see significant differences, within that, 1,500 to 2,000 cell range. If you're looking at a more heterogeneous population, if you're looking at a tumor sample, for example, then you obviously need to go much higher than that, But it's hard to give a specific number in terms of how many cells. It really is context dependent, but it does come down to how heterogeneous your population actually is. In this situation, the cells came from healthy patients, so we believe that they were actually representative of what we'd actually end up seeing in peripheral blood mononuclear cells. If you are looking at diseased patients, though, then obviously you're going to need more patients. And touching on the first question, how many samples you need is going to depend on how heterogeneous your population is. If you're looking for tumor populations, you're going to need more samples than if you're looking at the healthy individuals.
Okay, thank you for that response. The next question is, how many cells are, we asked that. There's a follow-up question to your if on 200 or so genes expressed per cell. This individual asked, if you can't measure splicing, how do you measure RNA velocity?
So every platform will actually have, will detect some portion of transcripts that haven't actually undergone splicing events. If you go through the paper that was by the Karchenko lab, they actually outline which platform has what percentage of the reads still retain introns. So they actually outline, for the different platforms, those sort of rates. And while you may not necessarily have, 60, 70, 80, 90% of all of those retained introns picked up in your analysis, from what is picked up, you still have the power to be able to make these calculations and make these predictions in regards to the cell fate. So to get a better understanding, I really recommend you actually go to that paper and go through and read what they actually did and how they were actually able to get the maps to model what is actually happening in the biological setting.
Okay, thank you for that. And the next question is, how many cells do you use for sorting? And how many cells did you obtain after sorting? I believe this might be a question for Chris Brampton, who can answer some of the flow cytometry questions related to this study.
Yeah, hi, thanks, Brandon. We started with about 350 million cells from the PBMC population. We sorted around 120,000 of those cells, and I think probably as we moved into the RNA-Seq apparatus after washing, etc., we probably lost 20,000, 30,000, so about a quarter So moving forward, we had about 90,000 cells going into the apparatus, and that was probably plenty.
Thank you for that. And a follow-up question to that question, how many genes are you able to analyze? And this is for Mahmoud, Dr. Bassal.
So how many genes were we able to analyze? So on average, I believe with the DD-Seq, you're able to get close to the 1,500 genes per cell mark. That's on average.
Okay, thank you for that. And another question, a follow-up question to how to measure RNA velocity. If compared to bulk set of cells, can you measure low and moderately expressed genes, or are you only measuring highly expressed genes?
So this is going to come down to the platform that you actually use, because the different platforms have different sensitivities. Generally speaking, though, you are detecting the highest expressed genes, moving down the scale to moderately expressed genes. You can actually detect low expressed genes, provided that you sequence deep enough. So basically, if you've got deeper pockets, you can actually sequence more and more and get that information from your single cell population.
Great. Okay, the next question is, why did you sort CD34 positive, CD45 positive HSPCs from PBMCs and not from bone marrow?
So that's actually a really good question. Obviously, it is easier to obtain the CD34 cells from sources such as bone marrow, but this work really sort of came about from Bio-Rad wanting to push the limits of the S3E, as they really wanted to sort for rare stem and progenitor populations at speed. So now that we've actually undertaken this work and we've actually seen some really interesting biology here, we're taking the work further. We've actually done some experiments looking at bone marrow, so we are expecting to see more abundance of the populations that we're actually looking at here. So you're definitely right. You can get more cells from bone marrow, but this really wanted to see how can we push the system to the limit and see what we can pull out.
Okay, thank you for that. And that looks like all the questions that we have. So again, we just want to thank everyone who attended the webinar. And if you would like more information on the S3E cell sorter, please follow the link that is present on your screen. And with that, we would like to conclude our webinar. So thank you again, everyone.